Okay, so welcome to chapter 33, and we'll be discussing and lecturing over an introduction of invertebrates. This will be a very, very long lecture, about like an hour, because we have 99 slides to cover. This is a lot of slides. And hopefully, we can get through this without any difficulties. But let's begin. So, what are actually invertebrates? So, invertebrates are animals without a spine. We are vertebrates because we have a backbone. We have vertebrae. So, us animals can be categorized as invertebrates and vertebrates. And remember that invertebrates have no spine, no backbone, and vertebrates have a backbone. Us humans have a vertebrae, and we belong to the phylum Chordata. Well, there is a subphylum. I forgot the subphylum. It's on top of my head right now. And I forgot to plug in my headphones. So, as we discussed, invertebrates are animals that lacked a backbone, and they account for 95% of known animal species. So, here we have the little tree. So, here we have our common ancestor of all animals, and then there's periphery, and these are our sponges. And periphery are basically one of the simplest animals of all time. But are they really classified as animals? Because they are not defined as triploblast or diploblast. Remember, as we discussed, the three germ layers, or two germ layers, is the mesoderm and the ectoderm. Those are diploblastic, and the triploblastic animals have mesoderm, endoderm, and ectoderm. The ectoderm gives rise to the skin or to the outer tissues of the animal, while the endoderm gives rise to the gastrovascular cavity, and the mesoderm gives rise to muscles and other organs. But sponges do not have any tissues. They don't have mesoderm, ectoderm, or any type of tissues. And they are sedentary filter feeders. Sedentary means they cannot move. They are just sessile. They're stuck at one place, and they're going to have to find a way to find food. And they are filter feeders, meaning that water goes through them, and whatever particles that are in the water, they capture it through choanocytes. So here is our typical sponge. It's a very simplified version. Well, it's already simple enough. So here we have amoebocytes. These amoebocyte spongiocele. A spongiocele is that cavity right here. So a spongiocele, seal means cavity or space. So think of this as a cavity. The osculum is the exiting portal. So here is the wastes all go throughout here. The water enters through pores. So these are our little pores, and it exits out the osculum. And we'll be discussing how these guys interact with each other. Because remember, have multicellular animals are multicellular organisms, meaning that they have cells that are different from each other, and those different cells interact with each other in order for them to live. They cannot live independently. They must depend on each other. For example, humans are composed of many different tissues, many different cells. There is neurons, there is skin cells, there is uh, epithelial cells, there is the immune system, which is a whole crap ton of cells, different types of cells with their own specialized roles. And if we took away even not even like one cell or a few cells, like 3 or 4, from our body. Like, let's say if we took away all, let's say, eosinophils, which is a type of leukocyte. Eosinophils cause inflammation and allergic reactions, the allergic reactions which is needed. But oh yeah, I don't. If we take away eosinophils, I will no longer suffer from allergies or allergic reactions. Well, eosinophils are kind of important because they attract other cells at the site of infection. At the site of the allergen or at the site of the invader. So it just calls all the other cells for them to attack the foreign invader. And if we remove eosinophils, who's going to call the white blood cells? Who's going to call the macrophages? Nobody. It's just gonna, that bacterium that's infiltrated your body is just going to go under the radar and kill you and take advantage of your body's nutrients. Remember that animals are categorized as multicellular and they all interact with each other because of cell surface proteins found in their membrane. So enough talking about ourselves, let's talk about sponges. So, hold on. So water enters through these pores, and these pores, I mean not these pores, the water is taken inside the spongocoel, which is the cavity. And the spongocoel is lined with choanocytes. So here's a choanocyte. This is the choanocyte. Right here is the amoebocyte. This is the amoebocyte section. But let's focus on the choanocyte first. So the choanocyte has these little collars. This is the collar, this little area right here. And these tentacles have mucus encoded with them. So when food particles enter the spongocoel, these food particles will be adhering to the mucus covered tentacles. And these choanocytes flap their flagellum, which moves down the food particles into the body, into more towards their body. And then the choanocyte will undergo phagocytosis, swallow that food, and then take it to the amoebocyte. It's kind of like a relay team. So this guy gets the stick, he runs it down to the amoebocyte, and he gives the stick, which is the food particles, to the amoebocyte. And the amoebocyte is giving those nutrients to the sponge as a whole. So remember that there's 2, 3, 4 different cells. There's about 3 different cells. There's the choanocyte, the amoebocyte, and the spicules. So there's about 2 or 3 different cells. They're all working together. And if we removed either one of those cells, the sponge will not function properly. If we remove the choanocyte, who's going to capture the food and give it to the amoebocyte? If we remove the amoebocyte, who is going to receive the food from the choanocyte? So each cell plays its own role. And it all depends for them interacting with each other in order to live and survive. So amoebocytes also manufacture skeletal fibers called spongin. So spongin is protein from the sponge, and then when a sponge dies, it leaves this protein behind. So it's just there. So Amoebocytes can also give rise to other cells, which is found in the mesophyll. So here's the mesophyll, but remember, this does not mean, not mesophyll, the mesohyl. This does not mean that it has a mesoderm. Remember, they don't have diploblastic or triploblastic tissues or germ layers. They're so simple. This is just a little structure they have. This is all they have, just a little amoebocytes and a little other different types of cells that I do not know about because I don't look, I'm not a PhD in sponges on periphery. But that is pretty much sponges in simple terms. We went over choanocytes and amoebocytes. They are totipotent cells. So totipotent cells are cells that can give a rise to different other cells. For example, stem cells, our stem cells can give a rise to neurons, muscle cells, what else? Other cells, liver cells, heart cells. They are totipotent, and they play roles in digestion and manufacture of skeletal fiber. So most sponges are hermaphrodites, which have both female and male reproductive structures in the same organism. So right here, let's say this guy right here had female reproductive structures, and this guy right here had male reproducing structures. So they go a thing called internal fertilization. Guess what it is? Fertilization inside a sponge. It's not that hard. So this guy will produce sperm cells and it would fertilize the egg inside this guy or this girl. And then what happens is once it's fertilized, the zygote is flagellated, which has flagellum, which means it can move. So this zygote will swim out of the parent and will go colonize a different area. That's how sponges move away. They move away from their parents. They don't want to be competing with other mature sponges at such a young age. So they want to go to an area where there's very little sponges or new areas with very little sponge diversity or sponge count or population so they can have the nutrients to themselves. The Venus flower basket or Eupicatella aspergillum is a glass sponge that builds its skeleton in a way that entraps shrimp-like crustacean inside for life. So, right here, these little pores, these little openings. So when a baby crustacean or a baby shrimp enters these pores of the sponge for a quick meal, because these sponges have food particles, like I said, the shrimp will eat the food particles and, enjoy its time there. And sometimes those shrimps would stay there for a few weeks or a few days, maybe a whole month, and it grows. Of course, it grows. Once it grows, it can no longer exit the sponge out the way it came from as a child because it was small enough to go through the holes or through the pores. But guess what? The shrimp does not care because he has infinite food from the sponge. And so he would continue eating and cleaning the sponge. So it is kind of a beneficial relationship between the sponge and the shrimp. And sometimes the sponge would have multiple shrimp in one area. And sometimes those shrimp have female and male shrimp. And when they reproduce, their baby shrimp are able to exit out the sponge via the pores the same way that their parents have entered when they were a child and go do their own little thing. So cnidarians are all animals except sponges because they don't have true tissues, and other groups that belong to the clade Emetozoa. So the phylum Cnidaria is the oldest groups in this clade, which includes jellies, corals, and hydras. So the basic body plan of a cnidarian is a sac with a central digestive compartment and the gastrovascular cavity. It is an incomplete digestive system, which means it has just a mouth, one opening for the mouth and the anus. So the anus is served as the mouth, and the anus is served, and the mouth is also served as the anus. So there's only one opening. They have an incomplete digestive system. And there's two different variations. There's two different body plans. The sessile plop and the motile medusa. Motile means it moves, sessile means it stays still. Doesn't really actually move, like go through one place to another. So here's a medusa. They can actually go through one place and another. So here is a simple body plan of the medusa and the plop. So the plop here is the body stock, and here we have the tentacles and the mouth and anus. Remember, incomplete digestive tract. The yellow means the endoderm. The ectoderm is presented by the yellow. Same thing with the medusa. The blue represents the ectoderm. The yellow represents the endoderm. And here is our gastrovascular cavity, which will be used as a hydrostatic skeleton, which we will discuss in a minute. And these tentacles of the medusa and the plop are armed with cnidocystis, used for defense and capturing prey. This is pretty interesting, so I would pay really close attention to this. Well, if you want. I think it's pretty interesting. I don't know about y'all. So, a nematocysts are specialized organelles with cnidocytes that inject a stinging thread. So this is how plop and medusas capture prey. So here on the tentacle, if we zoom all the way in, here we have little hooks. And these triggers, this little hook right here, this is a trigger. So let's say if let's say a little fish gets a little too close to the trigger and it accidentally brushes against the trigger. Once it brushes up the trigger, it is activated. Our harpoon is literally stabbed into the fish's flesh, and it's now attached into it. So this trigger is activated through a stimulus, whether through a quick movement or you brush it against really, really softly. Remember, these are really sensitive. And here we have the cnidocyte. The cnidocyst is actually the harpoon. Here we have the little thread, looks like a little rope, and it's coiled up. And once it's activated, the thread discharges, stabs into its prey, and literally has you latched on. And sometimes these nematocysts have enzymes and toxins to paralyze their prey. And once their prey is paralyzed, they will release digestive enzymes to break down their prey and feed off of them. So the gastrovascular cavity acts as a hydrostatic skeleton against contractile fibers, can work to cause movement. Remember that these medusas and jellyfishes do not have mesoderms, or they don't have mesoderms. So they don't have muscles for coordinated movements. They don't have a centralized nerve net either. They just have simple nerves. So, how do they move? So, here we have the gastrovascular cavity right here. These guys don't move a lot, but let's put it here. So, water enters through the cavity, the gastrovascular cavity, and it fills it up until it is firm. You know how you filled up a balloon, it's all firm? You can have you ever filled up a water balloon? It's flaccid and there is a firm water balloon. So once the gastrovascular cavity, it is completely full with water, the mouth will close to keep the water from spilling out. So pretend of the balloon, you fill up that balloon all the way you can, or until it's really, really firm, and you close up the mouth, which is the little opening of the balloon. Now, the contractile fibers now have something hard for them to act against, for them to act upon. Same thing with our bones. Our bones are structural supports. There's something hard for our muscles to work against. If there were no bones, we wouldn't move. Well, we would be able to move, but not as freely. But it acts as a hydrostatic skeleton, which gives the medusa structure and it enables them to move. And movements are coordinated by a non-centralized nerve net, which means they don't have a brain or ganglia. They just have simple nerves that are activated from stimulus and they move towards and away from stimuli. So medusoans. Medusoans include all cnidarians that produce a medusa. So a little jellyfish looking structure. And most hydrozoans alternate between plop and medusa forms. Drinking my coffee because I'm starting to get a little tired. So these guys are motile. So most hydrozoans, yeah, we discussed that. So let's talk about the stages of abedylia. I think I said that right. So here we have the main body structure of abedylia. I think that's how you say it. Let me Google that real quick. Oh, oh my God, I may have said that wrong. Abdulia. What the hell? What did I just get? I just got. I literally got mugshots of a girl. Obelia? Is that what's it called? Oh, it is Obelia. Okay, let me write that down. So, a life cycle of a Obelia. Obelia. So, the life cycle of an Obelia. So, here we have the structure. So, here's a colony of plops. And these plops have asexual reproduction and also sexual reproduction. So we have a reproductive plop here, and here we have an asexual plop, which is budding, which is clones. So they clone themselves as well. So here's a feeding plop, which means that, of course, it captures food. So the plop would detach from the colony, turn into a medusa, and these medusas have gonads, whether it's a female or a male. And let's say this is a female, and this is a male. So these are two different colonies. They have their sperm and egg. Their sperm and egg fertilize. They become a zygote. The zygote multiplies, multiplies, and multiplies. I mean, divides to become a larva or a planula. The planula becomes a developing plop, and the plop would go through asexual budding until it is a mature, and it will create its own reproductive plop, and it will start the cycle all over again. So here we have two different jellyfish, two different species of jellyfish. Let me erase this. So coastal jellies have a brief plop stage. So the moon jellyfish, right here, Aurelia, Arita, that's a beautiful name right here. And helmet jellyfish, Periphylla, Periphylla, which is an open ocean jelly, have no plop stage, while coastal jellies have a brief plop stage. And I want you to think about this. Why do coastal jellies have a brief plop stage and open ocean jellies do not? Let me give you a minute to think about that. So, simply to say that plops need a substrate, they need a surface for them to attach to. And a coastal, where the sea floor is really, really close to the surface, allows plops to stick to the sea floor because the coast is about, I don't know, 20 meters deep or 30 meters deep. While open ocean jellies, oh my God, like once you're in the open ocean, the sea floor is like 500 meters down or 100 meters down. And plops really do not have to travel all the way down to the sea floor and adhere, which is just a waste of energy and other things. So coastal jellies have a brief plop stage because they live in coastals because the sea floor is closer to the surface, where they have an actual substrate for them to attach to. The sea wasp, the Chironex fleckeri, is found in Australia and New Guinea and also around the Philippines and Vietnam. They are, like most box jellies, they have highly toxic cnidocytes. Remember, the cnidocytes is the harpoon, and the sting can lead to respiratory failure, cardiac arrest, and death within minutes. So I wouldn't like to swim in a pool or an area with these guys chilling. So let's move on to anthozoans. So anthozoans only occur as plops. So there's no medusa, just plops. They are made of corals and sea anemones. Corals have often formed symbiosis with algae and secrete a hard exoskeleton made of calcium carbonate. So corals, have you seen the Great Coral Reef? I haven't seen the Great Coral Reef, but they're pretty cool. These corals right here, once they die, they leave their exoskeleton behind. And then that serves as a substrate for plops and other corals to attach to. And then when that generation of corals and plops die, they leave their cytoskeleton and the cycle begins over and over and over, and that's how a reef builds, and of course there's other types of ways it can build. Now we're talking about worms and what's it called? Flatworms? Ocean worms? We're talking about worms and arthropods. And we're gonna take a quick break here because I haven't gone through this platyhelminths. I'm gonna do a quick break right now. It's like 5 minutes. Alright. Let's zoom back to our. Oh, I think I just read. Oh, I did the recording. I just wasted 5 minutes. Anyway, let's go over platyhelminths. And these are free living members of the phylum Platyhelminths. Live in marine, freshwater, and damp terrestrial habitats. Over half of the flatworms are parasites, such as flukes and tapeworms. So, yeah, it's over half. Also, it's over 50, so it could be 60%, a good chunk. And they are triploblastic and acoelomates. So remember, acoelomates mean they have a fake body cavity. It is not lined in endoderm, while a coelomate is fully lined, it's fully surrounded by ectoderm. And uh, I said that wrong. Sorry, it's the other one. Triploblastic. So a triploblastic, I said that, wow. If I was tested on that, I would have got that wrong. The triploblastic means they have a mesoderm, and the acoelomates means they have a gastrovascular cavity, but they don't have any other cavities, and it's completely filled with mesoderm. So it's basically like this. So here is our ectoderm, here's our mesoderm. And they just completely surround it. And here is our endoderm. It only leaves one cavity for the gastrovascular cavity. And they are coelomates. So this is called an acoelomate. Most flatworms have a gastrovascular cavity with one opening. Not bad, I sneezed. So flatworms are dorsally ventrally flattened bodies that shape all cells that places all cells close to water in the surrounding environment in their gut. So they are flat. They're like a thin sheet of paper. They're really, really thin. And try to think why they're so thin. Let me give you a minute to think. For gas exchange, duh. They're so thin that oxygen can actually diffuse through their whole body. So they can, and while they diffuse through their whole body, the cells can pick up the oxygen and use it. They're that thin that they can do that. They don't have lungs. Our lungs have alveoli, which are actually pretty thin. They're like really, really, really thin. So, actually, I'm not going to get into that. I'm just going to say, picture this is our alveoli. Here is our wall of the alveoli, and here is our wall of our blood capillaries. So once we breathe in air, all this oxygen, let's put a little dots, all this oxygen will diffuse through the thin layer of alveoli. It will diffuse through the thin layer of capillary blood vessels and oxygen our blood. So that's a very, 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 very simple of how we oxygen our blood through our lungs. So that is how our lungs work in a really simple term. And these flatworms, they are dorsal ventrally flattened for them to have gas exchange occur immediately. They don't have to breathe in air. All they have to do is just have contact with water, where oxygen is present. Like water just simply, not water, oxygen just simply diffuses. That's how thin they are through their whole body. So there's an excretory apparatus consists of photonephridia. So it moves water out the body and it maintains osmotic balance. So here we have the flame bulb, which is our little bulb that has this little cilia, and it's kind of like a turbine. So let me put it this way. So there's the tubules open on the outside of the body. So here we have the little tubules, and here's like little flaps or little openings. They move in. Water goes into the flame bulb, which have flagellum. They move the water through the tubules, and this actually makes the this actually helps regulate osmotic balance. So it's kind of like spinning the water, channeling it through the little tubule, and it's into the body wall, or into the body wall, or into the body cavity, or whatever they use, into their little gastrovascular system, or wherever it needs to go. So this maintains osmotic balance as well. Then free living species. Oh, and I think they do not have sections. I forgot to point that out. That platyhelminths are not segmented. They don't have segments, meaning that they don't have these specific segments as are arthropods. In arthropods, you see these arthropods, these little or polyplacophora. Oh, never mind. Little right here. Actually, I think I'm wrong. I think these guys have segments, so I'm not sure. That's a question. That's a good question I need to ask. I'll find out myself. Do platyhelminths have segmented bodies? Sorry I'm taking a while, guys. But let's go back to our slide. So here are free living species of platyhelminths. Planarians prey on smaller animals and on dead animals. They have sensitive eye spots and centralized nerve net. So they have somewhat of a brain. It's just more of like a mass of nerves. Not really a brain. It's called a ganglia. So they have nerves that run down their body. Here we have nerves. Here's our nerves, and they run down the body. And it means that these animals have more coordinated movements. They can control almost throughout their whole body. But remember that we are more sophisticated than these animals, because we have multiple nerves. We have multiple roads of nerves that go on the x-axis, horizontally, vertically, and other ways, while this guy only has nerves that go in one direction. But they do have directive movement. And the pharynx, here's our pharynx. Here is our pharynx, and here is the mouth opening. The pharynx, what it does, it spits out digestive enzymes on prey, and it sucks up the juices when waste are secreted. So the mouth is where it ingests all the good nutrients, and then it spits it out through the pharynx, while the pharynx also spits out digestive enzymes on prey. And these planarians are also hermaphrodites, and they can reproduce asexually or sexually through fission. So the cool thing about these guys, if we can cut these guys in half, two portions will grow into a whole organism. So it's kind of like Deadpool, Well, not really, not like Deadpool. Like more like, I don't know how you say it. You just cut these guys in half, and those two portions will grow. Each two portions will grow into their own organism. So parasitic species called trematodes. So trematodes are also called, schistosomiasis. are also called blood flukes, which live in the intestines and reproduce inside the intestines. So they parasitize a wide range of hosts and have complex life cycles with the alternating sexual and asexual stages. Trematodes that paraterize humans spend part of their lives in cell hosts. So these, where should I start at? Let's start at the snail host. So these snail hosts have larvae, ciliated larvae, and then when these snails typically live in rice fields where the water is not moving that much. Places without running water are more susceptible for germs and other nasty things. In this case, parasites, schisto, schistosomiasis, myiasis, and it will become a motile larva, and the motile larva would actually get into the hands of these workers, this human host, and that guy could ingest. Maybe he'll forget to clean his hands, or he puts his hand in his mouth after fondling with rice, and boom, you have one of these guys in your intestines, which they literally live in your intestines and feed off your nutrients. And the cool thing about these guys, well, they're not pretty cool, but the cool thing they have in that is that they have to evade two immune systems, and they do this by imitating protein host receptors and molecules that manipulate immune systems. So remember I'm not sure if y'all know this, but I think y'all should know this, that the immune system has receptors. They, let's say they have bodyguards. They ID check everybody. And these bodyguards, if they find somebody without the correct ID or a fake ID, they will immediately kick him out or kill him. Or that's basically our immune system. But these blood flukes would literally have a good, like a really well-made fake ID, not from the flea market, like a professional, like I'm talking about cartel grade. I'm talking about cartel grade IDs. They look so real that the bodyguard said, "Yeah, this guy seems legit. Let's just let him slide." And they just go under the radar. And it has to go through the human's immune system, which is more complex than the cell's host, than the snail's immune system. But they both function similar. They got some similarities between them. So this larva and this blood flukes have to evade two. And that's imitating protein host receptors and making molecules that manipulate immune systems. Now let's talk about tapeworms. Ugh, these guys, when I was a kid, I was afraid of tapeworms. Like, I was afraid of parasites, like worm parasites. But now I really don't care. So parasites are vertebrates, mostly, in parentheses, that absorb nutrients directly from the host intestines. So the cool thing about this is they have no mouth and they have no gastrovascular cavity. All because the reason why is because once they're in the intestines of the host, all they have to do is just absorb the nutrients. They just spit out enzymes to digest the. Actually, they don't. I don't think they spit out enzymes at all. They just digest your food on the spot. They just basically the food that you digested. They steal that food. They steal all that digested food, and they grow like that. So fertilized eggs produced by sexual reproduction leave the host's body in feces. So in areas with poor sanitation, or if you, of course, in poor sanitations, you are more susceptible in having tapeworms. In America, you really don't have to worry about this because we have running water and water treatment plants. Areas like in South America and in Africa and India and in wherever, probably like in Vietnam, I think, like in some Asian countries, they have poor sanitations. places with not a very developed civilization, they are more susceptible to many diseases which are considered harmless or treatable in our standards, in our Western standards as in Americans. Now let's talk about mollusks or mollusca. They're the most diverse phylum, well, second most diverse phylum. That's about 100,000 species. Most mollusks are marine, though some inhabit fresh water and some snails are terrestrial. Slugs are terrestrial. They are soft bodied and they are most protected by a calcium carbonate shell. So these are mollusks and annelids. We're halfway there. Actually, we're not even halfway there. We're like 30 slides there. Yeah, we're halfway there. We just covered mollusks. That's funny. But let's just go over these. Nematoda, yada, yada, yada. Okay, this is a lot to go over with. I might have to do part two. I might just, actually, no, I'm going to finish this all now. But let's not waste any time. We'll take a break at the 1 hour mark. We'll take a break, a quick break. Well, I'm going to take a quick break. I don't know about y'all. Y'all can just keep listening. Oh, I just lost my, I think it was 33. So mollusks. So all mollusks have a body plans with a muscular fluid, which is used for movement, a visceral mass, which develops into the organs of the mollusk, and a mantle, which develops the shell and secretes the shell. The radula, right here, is rasp-like, and it's basically the mouth-feeding structure. It's a structure that aids in feeding, and it scrapes algae on rocks. So it's kind of like a little rake. So a mollusk will go over a rock, and the mollusk will use its radula to scrape off algae, and they'll eat the algae. So here is the radula, and the radula is like a little rake. It just scrapes off, algae into the mouth, and it, digests. So it has a coelom. So guess what it is? It's a coelomate. So here's the visceral mass, and the visceral mass contains most of the organs: the intestine, gonads, heart, the gill, the stomach, and of course, there's the esophagus. Here we have the foot. The foot aids in movement, and the gills. And the gills is what is used for, of course, taking in oxygen. So, what? Why am I talking about hemolymph on my notes? Oh, because they have an open circulatory system. I think. Do mollusks have? I think they do, and I'm gonna go with that because that's what it says in my notes. That's the answer I came up with. I might be wrong, but I'm going to confirm right now. Let me see my phone. I think they have an open circulatory system. Do mollusks have an? They should have an open. Yep, they have an open. So they have an open circulatory system, and they have blue blood. Yes, I remember now. Have blue blood. So octopuses have blue blood. but I think they have a closed circulatory system, which means they have vessels and everything. While in snails and other animals that have an open circulatory system, they have blood literally flowing, literally surrounding the organs. There's no blood vessels. There's no channels for the blood to go through. Just pretend that the blood is literally suspending the organs and oxygenating them like that. So instead of hemoglobin, instead of hemoglobin, they use hemolymph. And these hemolymph uses copper, I believe. Let me search that up. So I believe I'm gonna go with copper. So this one, and copper gives the blood a bluish color. It's a blue-green color. So our hemoglobin has two sites. The hemoglobin is a protein. Not there's a hemoglobin found on red blood cells, and those hemoglobin have four sites. Two of those sites which binds to oxygen, and two of those other sites bind to iron. While in hemolymph, they bind to copper, I believe, and they bind to oxygen. So they have four sites, bind to copper and oxygen. So the hemolymph is the circulatory fluid and is pumped into the body cavities and sinuses. So it transports waste away from the cells. So remember, it kind of soaks the organism's organs and oxygenates them like that. They don't have any channels or vessels or veins. It's just all free floating. The life cycle of many mollusks includes a ciliated larval stage called a trochophore. Just remember that. I don't know why I wrote that down. So most mollusks have separate sexes with gonads located in the visceral mass, but many snails are hermaphrodites. Now we're going to go into Polyplacophora. Chiton, chitons, I'm going to go to chitons. Chitons are unsegmented, oval-shaped marine animals encased in the armor of 8 dorsal plates. They use their foot like a suction cup to grip rock and their radula to scrape off algae off the rock surface. So here we have the chitons. And here is the underside of it. Here is the foot. Here are the gills. And here is the head. I think that's the head. And there's nothing much about them. Just a little quick chitons. And we're going over gastropoda now. So 3/4 of all living species are mollusks and of gastropoda. And most are marine, but there are many freshwater and terrestrial species. And look, they have that dorsal ventrally flattened body, so that means they probably have no gills. And these, which means that oxygen just diffuses into their cells, and they probably have no gills. That's my guess. Gastropods move slowly by a rippling motion of the foot or the cilia, and they have mucus to aid in the movement. We've seen a snail and it has all that mucus. We've seen a snail has that mucus that leaves behind it. Well, yeah, it makes it easier for the snail to move across. So gastropoda with shells protects them from dehydration. And the gastropods live in drier climates. And in dry climates, the gastropods will seal themselves inside of their shell, making them survive longer periods of drought. So here we have a snail, and have you ever seen a snail like just a snail shell, and it's just empty. There's no snail. Well, that snail is probably bunkering down for the long run because there's zero rain because it needs moisture to live. You know. So what would happen is the snail would wrap itself into its shell, seal the shell, the opening of the shell with mucus, and lubricate itself to keep itself moist. And that shell protects them from the dry climate, the dry weather. And once rainier days arrive, once the rainy season arrives, or conditions improve, they will come out of their mucus enclosed home and they will continue. Oh, love darts. Several species of hermaphroditic land snails fire love darts into each other's bodies while mating. That's kind of interesting. So the moon snail. Ooh, predatory snails. They use their radula to drill a hole through the shell of other prey. So here we have a bivalve, which is a clam or an oyster, or a mussel or a scallop. And these guys will literally go on top of a mollusk. I mean, not a mollusk, a bivalve, a clam. Let's just put a clam. And they will drill a hole, a literal hole. Once the hole is breached, they will just start eating it. So once the bivalve is under the radula of this guy, he's pretty much screwed. So when you go to the beach and you find a seashell and you see a perfect hole or a little hole here, that means that you're just seeing a dead body, you know, the skull of a bivalve. But a bivalve is shells divided into two halves and are drawn together by adductor muscles. And some bivalves have eyes and sensory tentacles along the edge of the mantle. So here are little eye spots and sensory tentacles. The mantle of the cavity of bivalve contains gills that are used for feeding and as well gas exchange. And they also have mucus. It's mucus encoded to trap food particles. And the cilia will move those food particles into the mouth. Most bivalves are sedentary, and clams can pull themselves, and scallops can move by flapping their shells. So, pretty cool. So there's excurrent and incurrent bivalves. Excurrent means they expel water. Current means they take in water. So they take in water. This is an incurrent siphon, so it pushes out water. I mean, it takes in water, sorry. It takes in water. It probably uses this as to maintain osmotic balance. Then also to feed. It will take in the water, capture the food, and then spit it out to the excurrent siphon. Then the cephalopods, which means squids, octopuses, cuttlefish, and chambered nautiluses. So they're active marine predators with beak-like jaws surrounded by tentacles. And sometimes, what's it called? Tentacles and, I forgot. Tentacles, what else it's called? So here is the beak of a squid, or and I think squids and octopuses also have beaks. So toxins are present in their saliva. So this blue-ringed octopuses pose a major threat to humans because they have blue rings, and if you touch these guys, you'll die. And they're really, really small. They're like an inch. They're like half an inch. And the cool thing about these guys is that all octopuses and cuttlefish have some type of toxin. Some toxins are not harmful to us, but but they are harmful to their prey. But this guy, this blue ring, like I said, they're really toxic to us, and if we touch one on accident, or they secrete their little acids, or not acids, their enzymes or toxins, we're pretty much screwed. And the foot is modified. It has evolved to expel water and into tentacles, which are, as we know, an excurrent siphon. So the shell is reduced and internal or missing in most species except the chambered nautilus. So the shell is lost in mollusks, not mollusks, in cephalopods such as the squid and the octopus. But some octopuses use shells, of course. But the nautilus keeps their shells and preserves it. So all mollusks have an open circulatory system. Cephalopods have a closed circulatory system, which have blood. They have blue blood because globin uses copper. So they also have a well-developed sense organs and a complex brain. So here we have our little oxygenation map. So here are the gills, the site of oxygenation, where the oxygen diffuses. So here we have our blood with copper and oxygen. So here is our gills. It becomes oxygenated rich, and then those the hemolymph, I believe, I think it's called as hemolymph, gives the oxygen to the tissues for them to use it, of course. And then if those globin, those hemolymph, become deoxygenated because they gave up those oxygens, and they are oxygenated, they become oxygenated again, and then it starts all over again. So protecting freshwater and terrestrial. Nah, let's skip this one. So just remember that these cephalopods have a complex brain and develop organs, which makes them pretty distinct from other animals, other marine animals. I'm gonna take a quick break, a good 10-minute break, because I'm starting to get a little tired, but I will try to finish this as soon as possible.